No, you cannot.
Hello everyone, welcome to Lay It Out, the No Bullshit Science podcast.
With extra bullshit.
I'm Kiri.
And I'm Alice. And there's no Demelza. Kiri's very sad about that because Demelza's in San Diego with the rest of Kiri's research group. And Kiri's here with me.
I hate you, Demelza. I hope you're listening.
She doesn't mean that.
I do.
No, she doesn't.
Yeah, so the biggest conference in neuroscience is going on right now. It's every year, and it swaps between San Diego, Chicago, and Washington.
Oh, I didn't know that.
Yeah, and luckily for them, it's in San Diego this year, so they've all gone. But this conference is huge. It's literally like all neuroscientists around the world that go from every single field of neuroscience. So actually, most people kind of say that there's no point really going until the end of your PhD anyway, because it's so overwhelming.
Kiri's been chanting that in her head all day.
And you know the shitty thing as well? I think the year that I'm going to go, it's in DC. Which is, no, it's way less exciting than San Diego. Come on.
No.
San Diego is a beach town. DC is like fucking London.
You might get stabbed.
Well, I mean, more likely to get a shot.
Okay.
Yeah.
That's good to know.
Also,
isn't DC where they have the most crime?
No, that's Chicago.
Oh.
Chicago's murder capital of the USA.
Oh.
And the place I'm going to go live in for 3 months. I might not make it back, guys.
I mean, I was going to come visit you, but you're really putting me off now.
I'm sure there's like certain areas you just don't go into.
But that's like everywhere.
Well,
isn't it? Kind of.
Yeah, but here you're just going to get stabbed. You're not going to get shot.
True.
But you need to get in close range for them to stab you. Whereas if you're going to get shot, you can be anywhere.
Yeah, that's a fair point.
So I feel like it's safer here.
Okay, so I should say my goodbyes then when you're flying off.
Yeah.
Okay.
Well, I'm either going to get a shot or die from a heart attack from all the deep dish pizzas I'm going to eat.
Your arteries are going to look great.
Oh God, I'm actually going to come back so fat. It's not even funny. Guys, we had a really fun weekend. So we had our Halloween weekend. And there was a few of us, and me and Alice and this girl Laura, we all dressed up as dead Alice in Wonderlands.
We were actually supposed to have more dead Alice in Wonderlands. Everybody was supposed to be Alice in Wonderland, even the male attendees of the Halloween weekend, but then they were all a bit shite and bailed.
Well, so 2 of the males ended up being Mad Hatters, which worked quite well.
Which did work quite well, because it was like the Mad Hatters and all the Alices.
But maybe we should put a picture on Instagram of us as Alice's.
Okay, I'll get on that.
Because I did everyone's zombie makeup, and I'm not going to lie, it's pretty sick.
Kiri was like, if I fail my PhD, I can do that.
Yeah.
It was pretty cool. And the speed at which you managed to make people look dead was astonishing.
I made a very realistic bullet wound in like 5 seconds.
Yeah, that was probably one of the highlights.
Yeah. As I was doing it, I was even impressing myself. Like, I'd be putting bits of eyeshadow on and being like, oh shit, I am good.
So modest.
No, but it looks sick though.
It does look sick.
Yeah.
Okay, well, should I post the food first or should I post the zombies first?
Post the food first.
Okay. We had some great food. Oh yeah, we can talk about our food. Did we talk about our food last week?
Yeah. I can't remember.
We had some quite good food this week, but Kiri concluded she needed to put more lemon in her hummus, in case anyone's interested.
I made hummus and labneh and tabbouleh. And then I bought chicken and falafels and flatbread. Because I didn't have anything in my house yesterday, so I couldn't cook. Alice?
Sorry.
Can you do the Instagram afterwards?
Yeah, sorry.
Fucking hell.
I'm not all here.
But there was something I've learned about Alice.
You need to cut that out as well.
Considering, the girl doesn't lift heavy weights, right? But for someone like that, she is incredibly strong and floppy when she's too drunk. And so I spent half the night...
Both at the same time.
I spent half the night making sure that Alice stayed upright. And it was...
Stayed vertical.
Extremely difficult.
I mean...
she would wrap her arms around my neck and literally just pull me.
I was just seeing how strong you were.
No, she was trying to hug me, which ended up with her on the floor. And me on the floor with her. And this was on a boat, by the way. We're on a boat party. And I'm not talking big fancy yacht. I mean a little, almost kind of a dinghy.
It was not a dinghy. Oh my God, have you ever even been in a dinghy?
Okay, it was like a dinghy with floors.
Don't talk to a fisherman's daughter about a dinghy.
Okay, it was like one of those proper London boats though. It's not a big
It was a
boat
boat cruise on the Thames, but with zombies.
Yeah.
It
was good though.
Yeah, it was fun. From what I recall. Which
is not much of it.
Moving on. Should we bring on our guests or do we have anything else to talk about?
I think we're okay.
I don't
Let's
know.
do a quick.
Oh.
What?
So, I'm going to the Walnut Committee
yeah.
of California scientific meeting tomorrow. Yes, ladies and gentlemen, that is a thing. And it's in a fancy hotel in Hyde Park.
It is.
Yeah,
What
I'm
does-
actually really looking forward to it. I'm hoping the food's going to be good.
You know the people that decide to be in the Walnut Committee?
Yeah. They're walnut veterans. No, they're walnut heirs and heiresses.
Do you think, as a requirement, walnuts have to be your favourite nut?
I think by default they might become your favourite nut. Because they are pretty great.
I've only recently started to like walnuts.
Oh, they're nice. I like walnuts.
And I still
And they
don't
look like little brains, so they must be good for your brain.
Well, I realised I don't like nuts that look like brains, right? Like, I don't like walnuts and I don't like pralines.
Pecans.
Oh, pecans, that's the one.
Pralines are chocolates.
Oh.
You definitely like pralines.
I made pralines in Brussels. Anyway.
You've made them and you thought they were
Oh, no, wait, I
not.
watched someone make them.
Okay.
Yeah. I don't like pecans. But I've realised recently when I baked my cake that I actually kind of like walnuts, especially when they're toasted.
Oh yeah, they're delicious.
But I really don't like pecans still.
Okay.
But I like all other nuts,
Okay, that's good to know.
especially macadamias.
Oh, random. Most people are like, mad for pistachios or
But macadamias are so like
creamy.
buttery.
Yeah, they are.
They
aren't
are by far the worst nut for you.
Are they?
Yeah.
I should know that.
I think our guest is just having a giggle at the
Our
fact that I
guest-
keep
and
saying nuts.
I'm actually really impressed our guest is laughing silently. I didn't think that was going to happen.
Although she might pass out.
Maybe. Lack of oxygen to the brain.
Right. So what's happening at your committee things? Because this is actually a big part of what we do.
Yeah.
Yeah.
And I'm actually really excited because I don't get to go to these things very often because I'm quite niche. But
It just makes you sound so much cooler than you actually are.
I know, it's great. So yeah, I just go and there's gonna be loads of talks about like what's going on with walnuts and how great they are for your health and how we should all eat them because they look like little brains.
Sure.
Yeah.
I dare you to go say that to the Walnut Committee people.
I'm not gonna say that to the Walnut Committee people.
I dare you.
I doubt any of them are listening. Oh my god, you guys are great. You're so silent.
our
Our
guest
guest just
just-
silent sneezed. Silent, ladies and gentlemen.
Okay, so we're going to explain the format of the show today. It's a little bit different.
Just slightly.
We have no lay guest this week. Well, we have a wannabe.
We have an aspiring lay guest. He aspires to lay.
Yeah.
He's nodding in approval.
Don't we all
so-
aspire to lay? And also, Demelza isn't here today, so that's one of the main reasons why we got a science guest in. Because we know we need Demelza around.
We do. We need you, Demelza.
So we did have a lay guest, but unfortunately our lay guest couldn't make it because of some time commitments and changes of plans. But our science guest she's in neuroscience, but she's in a field that is still very, very different from what I do and extremely different from what Alice does.
Indeed.
So, yeah, and then our wannabe lay guest was also going to talk about his work, but we're not sure. Do you still want to? He'll talk about his work as well.
Yay! I'm extremely
From
curious.
his previous past non-lay life. Yeah. And then he'll go back to lay mode for
the paper.
our science guest.
Yes. We'll see how good he is at being
So we
being
have Rachel and Tristan.
Hello.
Welcome.
Hello.
You can talk now.
Yay!
It was
So we
hard
always-
being a ghost.
Yeah, I ask, or well, you normally ask, but where I was like, is it hard to not talk? And I don't know, I'm kind of like, I'd like to sit back and chill. But everyone's always like, no, it's so hard. Was it hard?
I was just laughing a lot, and I was like,
Yeah.
I normally want to, express
Yeah.
vocally my joy at all of the funniness. But then I was like, and that sneeze hurt
That
me.
was I'm not surprised. That was like so silent.
Yeah, that was good.
The most silent sneeze I think I've ever not heard.
I think you've actually been our most silent guest.
Yeah.
Today.
You know, you could have actually let sound out while you laughed.
I couldn't.
No,
No.
you're just too
Professionals.
dedicated.
You explained the rules, and we had to stick to them. We're very good like that.
I'm afraid of failure. That's why I'm a PhD student.
I feel like if you're afraid of failure, you definitely should not do a PhD.
Please don't tell my supervisor.
So actually, with Rachel, so me and Rachel, we don't actually know each other that well. We've only met twice.
Yeah.
And we met on a course about how to teach graduate students.
Preparing to teach.
And then we met at a neuroscience debate, which was incredibly depressing. The question was, does academic neuroscience have a future? And we both said no.
No, of
Maybe
course
we'll
not.
save that for another
Silly
episode.
billies.
But actually, so I met Rachel before we'd even started recording the podcast. And she was actually one of the first people that I messaged Alice and Damaza saying, guys, I've met this girl on a course. She's fucking brilliant. We need to bring her on as a guest on the podcast.
You did. I remember that.
Yeah.
That's so nice.
And
Then, so I spent a lot of time on the Waterloo campus, and
that's where I'm based.
I was walking into the building, and then I see this
And I think I
face.
was with you,
Yeah.
potentially.
I see this face outside the building on a plaque, on like a, what is it, like a...
Just like a
sign?
glass panel.
Like a little...
like one of those like
motivational quotes.
Yeah,
with
it
the
is a
picture.
motivational quote.
And then I looked at it and I thought, oh my god, I recognise this girl. And Alice was with me. I was like, Alice, this is that girl that I said
has to come on the podcast.
Yeah, that's me.
I see you every day, sometimes twice
It was nice to
a
meet
day.
you.
I feel like I've known you for the past 11 months.
I don't know what to say.
Precisely.
One, how did that happen?
Okay.
2, how does it feel?
Okay. Number
3. Are we the first people to mention it to you?
one. I'll go with 3 first. No.
No, I wouldn't have thought so.
It comes up a lot. And it's especially with
You
when
famous.
it's like, yeah, a little bit. Like other professors and lecturers and PIs in the department. And they'll come to me like, Rachel, I've seen you. You look so familiar. It's like, yeah, that's me. Yeah, I'm on the building.
Hang on. Oh, wait. 4. Is that picture anywhere else?
Oh my
Were you
God.
just on
Yes.
Waterloo or were you on other campuses?
I'm in Bush House.
Oh my god.
Bush House is the fancy
Bush
one.
House
Yeah.
is so glam.
What is Bush House?
It's the new...
I've never heard of this place.
Yeah. Go on.
It's not what you think it is. It's the new Strand building.
Oh.
It's shiny. It's
and
like-
you're in it.
Yes, she
course.
was there before I was there, obviously. So how did that happen? Well, when I finished my undergrad, I spent a year basically as a full-time student rep. I was one of the student officers in the Students' Union. And my role was Vice President for Welfare and Community. And so I was involved in pretty much anything to do with being in a liberation group or a minority or anything like that. And because I tick a lot of boxes, basically I worked on getting the university to support BME students more, so Black, minority, ethnic students. And part of what we did was, it was all in this project called Open Doors, and that's what all of the photos are about. It's like inspiring BME students, and they asked me if I'd be happy to be part of the campaign, and I was like, yeah, sure, you can take my photo. Like, who's gonna look at it? And then
It turns out a lot of people are looking at
and not
it.
even like the windows that on Strand that they change now every once in a while. It's like, I'm on there.
Yeah.
No,
I'm not going.
You are on the building. Like,
Yeah.
I think there's pretty permanent looking nails in that board of glass. Like, they're not going anywhere.
Yeah, that thing will be the only thing that has been at King's for longer than I have. By the
And
time I
yeah,
leave.
do you know what? I'm gonna see you every day for the next 3 years.
It's part of my campaign to really just up my profile. I think.
Yeah.
I'm hoping that gonna get some followers through this. Maybe I'll do some viral marketing later down the line.
You should have put your Twitter handle on there or something.
That's the way to go. I think next step I'm gonna have to deface it.
I mean, there's nowhere else. There's nothing else you can do, really,
No.
is there?
Do you know what?
How are you gonna top that?
I have a quite
no.
serious question.
Please.
I have actually quite a serious question about the BME thing. So you were like the BME rep person, right? Okay. So a few months ago, actually it was okay, whatever, some time ago, I was going to go to this BME Women of Science event thing. And in my head, I was like, yeah, I'm going to go. It's going to be great. I'm going to support it. And then I was a bit like, actually, hang on. I don't know if BME Women in Science is actually going to be, well, like, not welcoming, but inclusive of an Asian woman, because actually we have no problem in science.
Okay.
Good question.
So, I would start off saying that, I mean, do you feel like you have no problems in science?
Not as a woman, but as an Asian.
Yeah.
Like, there's definitely a lot of us in science.
So I feel like from what I've seen and what I've read, the issues are different but can still have an impact. So the stereotypes, obviously, you know, every group of people faces a different set of stereotypes. And for black people like myself, it's that we are either lazy or ignorant or not smart as the rest. And that, you know, makes itself known in a bunch of different ways. But when I read into it, and especially when I was reading about postdoctoral researchers, so after PhD, like for Asian people in general, so South and East basically, the stereotype was more that they are hard workers and dedicated and can just keep going and going and going. And obviously that would give a different pressure to anybody who feels like, maybe I'd quite like to take a break right now.
Yeah.
So it's more that the issues are different, and maybe like kind of more this kind of discrimination we're used to seeing gets spoken about more. So it's almost like two sides of the same coin.
Oh God, I could talk to you so long about this. I have so many opinions on shit like this, because I'm always like, yeah, it's just different. Like I feel like that pressure can be just as damaging.
Yes,
as
definitely.
not being given the opportunities in the first place.
Right, exactly.
Totally.
But anyway, cool. That's all I wanted.
Yeah.
Let's not turn this into like. That's a whole different episode. That's like a whole. That's a whole separate podcast.
Yeah, I think so.
I could literally talk about this forever.
That's your Black History Month segment. So
Oh my God! Now we can use hashtag Black History Month.
That's exploitative.
Okay, that's really interesting.
Yeah.
And yeah, no, the minute we saw your face outside the building, we were like, we need to talk to her about your face outside.
Yeah. We always have, on the day of the podcast, we always have this little, what are we going to talk about today? And it was like, what are we going to talk about? Halloween. Rachel's face.
The next time you're at Waterloo, you should just snap a pic, post it on the Instagram.
Yes. Oh my God.
Oh boy.
You're giving me all the Instagram ideas.
There we go.
Look at me go.
So Tristan,
Yes.
aspiring
No, I'm not aspiring. I am a layman, guys.
He's an under.
Everyone is lying to you right now. I'm a layman. I'm as lay as they come.
So tell us,
Too late to function.
what have you been doing for the past 5 years?
A PhD.
For those of you that may not have heard that, he said a PhD.
Yeah. But before we get into that, because that's going to be our science bit.
Is it? Oh God. Okay.
How do you and Rachel know each other?
Oh yeah.
Well, how?
How do we know?
It just feels like we've known each other for so long.
We pretty much
You just
have,
found each other.
Yeah.
Yeah, so
It was fate.
As an undergrad, I was a bit extra and basically
Oh, really?
begged.
Surprising.
Never. You got to be extra if you want to be on the front of the building. That's what I'm saying.
To all you young people out there with aspiring dreams to be on a building,
Be extra.
Rachel's insider advice.
Hashtag be extra. So I begged my personal tutor for some lab experience in the first summer of my undergrad, and then from then on,
You are one of those.
She's on a building. Of course she's one of those.
anxiety can take you to amazing heights.
I think that might be our episode title.
This is a common theme now by this point, isn't it?
What, anxiety?
Yeah.
Oh God, we're all gonna have like mental breakdowns by the end of
What do
it.
you mean?
Oh, we have mental breakdowns like every other episode. So
Yeah.
it's fine.
There's always one of us is having a bit of a weird day.
a weird day.
But yes, Rachel, you were being extra.
Yeah, I was being extra, so I was in the lab, and Tristan was in the lab doing
I
PhD.
think I just started my master's.
PhDing,
Masters.
being lay.
laying back.
It was a very laid back PhD. It was very, very relaxed. I didn't cry in the stairwell at all at any point.
If you don't cry in the stairwell, are you even
Are you even doing a PhD?
But yeah, we were
Yeah.
young, we were dumb.
Oh my god, we were so beautiful back
We
then. No, we are
were
still beautiful now. But yeah, we were a lot younger.
Yeah, so doing science basically.
This is how we know each other.
So wait, how old are you?
Oh god.
57.
I'm 21. Is that believable?
It actually is.
Oh my god.
That's
yes.
the thing, though.
Yes. No, I'm 27.
Okay.
Yeah. So I've
So yeah,
done some time.
it's all right. We
That's,
are
you're quite
nearly
young
as
to
old as
finish.
you.
Or
Yeah.
well, you are quite young to finish your PhD, are you?
I guess. I don't like to tell people I finished my PhD because I'm a lay person, everybody. No, I don't think so because I think a lot of people go into their PhD kind of straight from undergrad
Yeah.
now. So they,
Yeah.
I think a lot
I
of people
suppose it's getting younger. Like, if anything, we're old to start.
Yeah. Well, not really old, but like the older end of young.
Yeah, like I know more.
It's either us or then like mature students.
Yeah, I know a lot more people now that are probably younger than me, but at the same
Yeah.
stage. Then
How old are you?
I'm 26.
As am I.
Oh, did you want to guess?
Yeah, well, no, I was going to
Ah.
ask, but then I was like, is that rude? I just don't know anymore.
Yeah. How old are you?
24.
You're
Oh,
a
you
baby.
Such
Thank
a
you.
child.
Love it.
But yeah, I mean, like I think a lot of people I meet now who have just started, who are doing their PhDs are in their are 24.
Did you go straight from undergrad? No.
Well, I took the year out to
Took a year out.
yeah, get on the wall.
I think it's
No.
getting more
Oh,
and
yeah.
more normal to
Focus
just
on my goals.
Yeah, exactly.
Getting on
Yeah.
the wall.
Especially now with the integrated master's PhD
Exactly.
programs cropping up all over the place. It's like just straight after undergrad, go.
Yeah,
PhD.
yay
Go, go, go.
guys, PhDs are not special anymore.
Which I don't know is well, I guess it's a good thing, but I feel like PhD is such a commitment. I don't
Yeah.
know if undergrads are just finishedly recent undergrads are ready for that kind of thing.
I
Yeah.
absolutely agree. I say
I
this
agree too.
all the time. Like,
Because I feel like you come out of it with an attitude of, okay, what am I going to
Yeah.
do next? And I don't think that's the right attitude to go into a PhD.
I don't think an undergrad could have prepared me for how intense the master's was, and I don't think the master's could have prepared me for how intense the PhD was going to be. And then I think I needed some extra life experience to then get me this far through my PhD.
Yeah.
I don't think I would have been anywhere near ready if I'd come straight into this.
I think that's the thing because people kind of ask, do you regret, doing a PhD so young or whatever? Or do you regret taking a year out to do something completely different? And it's like, no, that's pretty much the only way I was sure that I wanted to
Yeah.
do a PhD. I
Yeah.
didn't know if I wanted to spend all of my time campaigning and changing things and maybe I liked science like on the side or if I really love science, but I was also passionate about making the world a bit less shit. So yeah, I think having the chance to explore that has definitely made me more focused on my PhD and just happier with the decision when things get rough as well.
Yeah.
But,
Yeah, I always tell, whenever people ask me about advice or what they should do, I always tell them that taking some time out was the best decision I ever made. Like, I've taken 2 years out in total at different points, but they were the best decisions. So I took a year out before my master's and then a year out before my PhD. I worked for one of those years. But the other year I just fucked about for a bit and it was honestly the best decision.
Yeah.
I was so glad I did it. Because also I was kind of against gap years a bit. Because I was like, I just want to get it done. I want to get it through. But then things didn't work out and I fucked about some more with my education. Had to take a year out.
So what you're saying, it was an unintentional year out.
Yeah.
But it worked out. It all worked out for the best.
Yeah. But I'm
On the
so
other
glad I
side
did
of that, though, I took some time off, but I didn't know what I was doing afterwards, and it stressed me quite a bit.
it. Yeah.
So I think there's different aspects to it.
Yeah.
While I'm glad I did take that time, I
Yeah.
was quite nervous about the future, but maybe that's just me.
Yeah, I didn't take any time. I just finished my undergrad and then went straight into the program, and this is why I'm a lay person now, aspiring. But yeah, no, I definitely recommend don't just jump into a PhD. You gotta know you want it, and
Yeah.
I think you need time to come to
I
that
agree.
decision.
Yeah.
And also take breaks during your PhD too.
Oh yeah, don't be afraid to do that because I feel like a lot of programs just let you pause. And if you feel like you
Yeah.
need to do it, because
Yeah.
your mental health is important,
Yeah.
guys.
Yeah.
Dude.
And you actually come back feeling so revived,
Yeah.
I find.
Dude, I'm over a year into my PhD and my first real holiday is at the end of this month.
Oh man.
No, at the end of November. And I would not recommend anyone do this.
where are you going?
Morocco.
Oh my
I'm going to Marrakesh.
Oh,
It's gonna be really
amazing.
But yeah, that actually did take a massive toll on my mental health. Yeah, I wouldn't recommend it at all. take some breaks.
How long are you going for?
Just a week. But it's my first all-inclusive experience. So I've always been a bit snobby and turned my nose up at people doing all-inclusive because I'm always like, why would you go all the way on holiday just to sit in a resort and not explore anything and blah, 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 blah. Such a waste of going on holiday. But actually, this first year of my PhD was so intense. I was like, I get it. I want one.
Treat yourself.
I want to not lift a finger.
Yeah. But, so we're going to do a few day trips anyway. But I'm looking forward to it.
Lovely.
Should we talk about some science?
Yeah.
Oh,
Yeah.
Okay, fine.
Should we
I
get your
think
bit
there
out
was
of the
a-
way?
Yeah, let's do that.
What?
I thought there was something else I wanted to say. But now I can't remember. Okay, moving on. Science.
Okay, yeah, so back before my life as a layperson, I was doing a PhD. And so,
Before my real life started.
shall I, I was going to say, shall I say what the title of my thesis was? Like, I don't know if I
Yes.
can even remember. Oh. Okay, no, I can. Okay. It was cerebellar development. No, that's not right.
Those were 2 key words.
You are just so good at being a layperson.
I know, right? Look
You're
at
just-
me.
you're
I'm
just-
just the best layperson ever. Okay, no. It was cerebellar development and the origins of foliation in the no. Oh my god.
Okay, question. What's foliation?
Yeah.
Okay, foliation is basically folding.
Okay.
And my PhD was looking at the folding pattern of a specific part of the brain called the cerebellum.
When the fetus is developing.
Yes, but so I was looking at other animals to kind of extrapolate from those models what might be happening in
animal
humans.
research warning. We're going to talk about animal research from both of our guests. If you don't like it, go away.
Oh yeah, yes.
There you go.
So yeah, no. My thesis was basically looking at the cerebella of different animals which had different degrees of folding in their in the cerebellum. And so I was looking at alligator brains, snake brains, and turtle brains.
Oh shit.
Yeah, and I was just trying to figure out how those came to their shapes, and if there were any kind of underlying molecular mechanisms that could then be applied to the humans.
That is so cool.
But what is the significance of the folding?
So the folding basically increases the surface area
Okay.
of the cerebellum and gives it a lot more computing power.
So what makes
Okay.
it's what makes us the reigning species because we've got bigger surface area of our brains.
Oh shit.
Yeah.
So that's why we're so smart.
Yeah.
Exactly. Although, so the cerebellum
So the more
is...
foldy your brain is, the smarter you are.
Yeah.
Oh.
But, the cerebellum is the part of the brain that's kind of in charge of, like, fine movement and, like, balance. And I am of the opinion that mine doesn't really work very well.
Mine doesn't work very well either.
Oh my god, yours definitely does not
I
work well.
I fail sobriety tests when I'm sober.
Oh my god. Why are you doing sobriety tests?
I'll tell you about it afterwards.
This is for a later episode.
Yeah. I'll tell you about it afterwards. We can't talk about it on the podcast.
Yeah, so that's my thesis in a nutshell, I guess.
God, there's a lot of nuts in this
Nuts.
podcast, isn't there?
So does if your cerebellum is about like fine motor control and balance and all that kind of thing, do people like trapeze artists or circus acts have a more developed cerebellum?
I don't believe so, because the cerebellum kind of, like many parts of the brain, it kind of just stops developing after you reach maturity.
Not the hippocampus, guys.
Like many, not
Hippocampal
all.
neurogenesis.
But yeah, so once your cerebellum has kind of reached its like optimum folding, which happens kind of like early in life. So when you're a baby, maybe 1 or 2 years old, it's like that for your entire adulthood, basically. And maybe trapeze artists have some kind of better, finer tuning within the cerebellum circuitry, but the folding itself stays the same.
Okay, I have so many questions about the animal brains. 1, where did you get them from?
Oh my god.
2,
There's
How big are they?
How big are the brains?
The brains.
Like this small.
Oh my
Okay,
god.
this is a podcast.
So
What coin?
he's indicating about an inch, I would say.
Yeah.
Yeah.
Even an alligator brain is that small.
Well, I was dealing with alligator fetuses.
Oh,
So
okay.
it's that small. I
Okay.
guess it grows much larger, but I did not have access to.
So, sorry.
Although we did, well, okay, we weren't looking for big alligator brains, but we did because I went to Florida to get these brains. But
Oh my god, you got to go to
I got
Florida.
to go to Florida. It was amazing.
People get to go such cool places for their PhDs.
Especially if you pick evolutionary developmental biology. What was I saying?
You went to get the brains.
Yes, I went to get the brains. So I just worked with embryonic samples, and the lab in Florida kind of gave me these eggs. And then we went on like this nature hike along the Everglades, or I don't know where it was, somewhere, natural plains of Florida. And we were walking along, and this alligator just like comes up to like the side footpathy thing, and it was like 2 meters away from us, and we were like, oh my god, shall we take a selfie with it? Don't take a selfie with an alligator.
Oh god.
Because the minute we tried, like we turned around, turned the camera on, and then we were smiling, and then the alligator just starts like running towards us, and we're like, run!
Oh my god.
But yeah, so that's just a funny anecdote from when I was in Florida collecting brains.
Are alligators the one that spin you till you drown, or is that crocodiles?
I don't know. I've never heard of this.
It's one of the big reptiles will bite you in the crotch and then drag you underwater and then spin you, and that's
specifically in the crotch.
I think that's usually where they grab you, and then that's how they kill humans.
God.
Whereas the other one does it in a different way.
That's making my skin crawl.
No,
Sorry.
Another reason to not try to take a selfie with a large crocodilian reptile.
So then, what did you find, or sort of how do you then relate that back to humans, and what are you looking at specifically?
Well, I think one of the main findings of my thesis was that there's different patterns of cell division during cerebellar development, which affects the final foliation or folding. So in the turtle, so imagine like this, oh my god, I was gonna say imagine, but maybe I'll just try to explain it more simply. So in the turtle, which doesn't have any folding in its cerebellum, the cells that make up the cerebellum only divide at the one edge, so it just kind of elongates. But then in humans and birds also have a very folded cerebellum.
They need to have quite fine motor control.
Yeah.
For the wings.
They fly and shit. So. And then, in mammals and birds, during cerebellar development, the cells kind of divide across the entire tissue, across the surface of the entire tissue, and that leads to these mounds of folding. So that was like the main finding of my thesis. There's others, but...
Okay, so every paper that me and Alice have talked about so far have been quite clinically relevant, or like obviously clinically relevant.
Yes. Yeah.
So
this is not that.
Yeah, so to then relate it to, I think these are things in science that people then start to kind of not really get why we're doing, right? So for people listening, how does what you're doing then, and Rachel, you as well, how does that actually, why is it important? 2, how will it help humans clinically? And 3, I don't have a third question.
And 3, all questions need to come in 3s.
We'll think of a third question to answer while we're formulating our answers. Do you wanna, or should I?
You can both.
I guess it's interesting because I remember hearing a story of a woman who's born without a cerebellum, right?
I thought of a third question.
Okay.
Okay, third question. What motivates you to do developmental research, considering that a lot of motivation for scientists is that the clinical stuff helps in the end?
Okay.
Yeah.
Well,
Or has a direct relation to disease, sorry.
Well, a lot of so my research specifically deals with cell division, and a lot of the things that happen during early development are the things that go wrong later on that give rise to cancer. So that's, well, they're very far removed, my research and cancer, but that's the thing with basic science.
You have to start somewhere.
Yeah, you have to start somewhere, and when you're looking into these very specific niche topics, you don't necessarily think about how they can relate to clinical studies and how they'll benefit humans, but eventually all of that knowledge is just going to trickle down,
Yeah.
and it's going to help us understand more about ourselves and help us with treating diseases and stuff like that.
I think for me, the interest of basic or pure research rather than clinical research, I just think that the brain is one of the biggest, if not the biggest, unsolved puzzle in the world. And I'm obsessed with puzzles. Like, anybody who knows me knows that I do at least 1 to 5 crosswords a day, because I freaking love puzzles of all sorts.
Like,
I'm like,
if you go home with Rachel, if you like have the pleasure of commuting with her sometimes, like I do, she will pull out like some newspaper from somewhere and be like, crossword now.
yeah. Sometimes
I need my daily fix.
It's actually an addiction, but I'm really good at them now.
Sounds like an addiction. Oh my god, shit, you must have a really good vocabulary.
Yes, I do. It's not up for discussion. It's just
It's
true.
just a fact.
Yes. But the point is, I've always kind of been obsessed with riddles and like those books when you get as a kid and you have to like solve by, you know, deduction. Who was the murderer? And it's like the person sat next to the scissors or something random like that. But kind of the clinical stuff, to me, you look at it and it makes sense. But there's so much in the brain that we don't even know how to look at it in the right way to make it make sense. And I guess it's almost like if you look at the stars and you can track them moving and you can think about theories about how the universe works, but you know, you would never look at the sky and see a black hole. And to me, it's like we don't know so much. I would just love to know more that we can't, that isn't obvious kind of.
You can't just see.
A hospital, wherever. Exactly. That's why I think.
Yeah, like the most annoying thing about our field is that we can't just take someone's brain and look at it.
Yeah, exactly. So that's why I find this stuff super interesting. And that's like no shade to everyone
It's alright, it's not taken.
anyone some of these obvious questions. Wait for a riddler like me.
But I mean, okay, so then...
I mean, whatever gets you out of bed in the morning, you know?
So then for our audience, I think we still need... the question, unanswered question is still though, how does this apply? Like, you know, what's the point?
Well, you kind of answered that a little bit.
Yeah.
Like, it trickles down.
Oh yeah, sorry.
You did.
And then so like the genes that you like, the genes that you you find out are working in these basic pathways.
Can be targets.
Yeah, they're the same genes that become mutated in cancers and other kind of diseases. And it's just, the more we know about those kinds of things,
The better.
It's better, right?
Yeah.
It can only be a good thing.
Exactly.
Yeah.
Exactly.
And when people talk about the miracle of life and like the odds that you would be born and all the things in evolution that led to that, it's like if you understand every step of how the brain develops, like the luck doesn't stop when the sperm meets the egg. Like there's so much that goes on during that time that we don't understand.
And there
that
is
maybe
so much we don't understand.
Yeah.
And we can't cut up a human brain, like you said, so we have to cut up other animals.
No, developmental stuff is super interesting. I remember during all my lectures in developmental neurobiology, there's been some crazy experiments though that led to actually showing how everything's done. Like
the two-headed frog experiment.
yeah, that one. That one's
was
crazy.
What happened?
So they, during, oh my god, I'm going to try to explain this, but I don't know if I have to remember. But Rachel, you were in an embryology lab, so you'll be able to help me out. So during development, when it's like you're basically just a tiny ball, there is this organizer region that kind of coordinates your axes. So it says that this is going to be your back, this is going to be your front. And then, so what they did was they took one of these organizers from another embryo and stuck it on an embryo, so it had two.
Oh shit.
And then so that embryo kind of developed with two axes, so it grew two bodies fused together.
Where were they fused from? The back or the front?
the spine,
Yeah,
right, is
I
almost
think
like
so.
a Y shape, kind of joined in the spine.
It's crazy.
Yeah, developmental biology is cool, guys.
Yeah. I did one module in, I think it was called would this make sense? Developmental embryology?
Absolutely,
Yeah.
yeah.
Perfect
Embryology.
sense.
Yeah.
Wonderful.
Yeah, cool. I did a module in that in like my first year of uni, and it was like German. Like, I was like, fuck me. But like, it's one of those things, you know, like I had so many modules on like research methods and statistics, and I was like, fuck this, I hate it. But they really do come back to you. That module, even though while on first sight may not fit into that bracket of, oh, I hate doing this, but it actually will help down the line. It really does. Like, sometimes, even when you said when you're just that tiny little ball, I was like,
Yeah.
blastocyst?
Yeah.
See, here we are. That's my whole module. Was weighted on this moment.
Oh my god. Do you remember who your lecturer was? You should email them and be like, it's paid off.
It's finally paid off. 5 years later.
See, this is the lesson, kids. Stay in school.
It really is true. Just listen for like 5
I mean,
minutes.
I've never used trigonometry in my life, so
Lying.
you never know.
You probably use it in ways you don't even realize.
Although, I will never forget a Sokotoa.
Yeah.
Yeah.
Do you
Like
never
the
calculate?
trigonometry calculations to help you remember which ones you need to divide by each other.
Sine.
Sine.
Cosine.
Cosine.
No, but Sokotoa is
Sokotoa,
like
sine,
some opposite
opposite.
over hypotenuse.
Yeah.
Yeah, and then cosine is so ka what's that? What's A?
Adjacent.
Adjacent over
Hypotenuse.
yeah. Anyway,
And then toa opposite over adjacent.
yeah. Tangent.
Don't, yeah. Yeah, time.
Anyway,
But yeah.
Moving on.
We never used
just
that,
used
but we
it.
remember it. Yeah.
We're bringing out all the old school bangers today.
You used it to bond. We bonded over
did.
our shared experience of
trigonometry
trigonometry.
brought us together, guys. Okay, cool. So then, Rachel, do you want to tell us about the work that you're doing?
Sure.
Yes, I'm a layperson again.
So when I met Rachel, the reason why I was like, we need to bring her on, apart from her lovely personality, was the fact that she works with Zebrafish.
Yeah, I love my fishies. Me and my fishies are best friends.
Guys, I don't even know what a zebrafish looks like.
Okay.
Should I Google this right now, or are you going to paint me a picture?
You can Google it, but
But paint a
I'll
picture.
paint the people at home a picture.
Do.
So
Maybe I'll put it on the Instagram.
There's too much Instagram.
so
I'll put it on the Instagram when the episode gets released, because otherwise there's no context.
yeah, that's nice.
You're welcome.
So a zebrafish is a tiny little fish. Well, it's not super tiny. It's about 1.5 inches long. And the reason it's called a zebrafish is because lots of the varieties have little stripes on their bodies.
Cute.
So they're the types that you can see if you go to a pet shop. And in fact, if you go to a pet shop and you want to buy a glowing fish, that is going to be a zebrafish that has been genetically altered in a way that's similar to how I use it in my work. So yeah, they're tiny, they're cute, they
They're
live
cute.
in really nice places on Earth, in like, where is it? Like South Asia?
Yeah.
Yeah, South Asian little fish. So they're cute and wonderful. Do I need to? Some of them have really long fins. Wait, maybe I should stop talking about fish and start talking about my PhD.
Are they not the same thing?
Yeah, well, yeah, basically.
That was a really good description of Zebrafish.
Yeah.
Yeah,
Good.
I like it.
I'm happy. So, in my PhD, I'm looking at a part of the fish brain called the optic tectum. And tectum is a Latin word and it means roof. And basically, they're two big lobes that sit on the top of the zebrafish brain. Yes.
Is it the same origin as a tectonic plate?
I believe so, yeah, because that would be like the roof kind of surface of the earth. I would assume so, but I don't
Look
know
at
for
me.
sure.
There you
I'm
go.
going to trust you on that.
Or maybe I should do crosswords.
You should. All right, I'm going to assign you a crossword.
Oh God, don't do that.
2 weeks' time.
Oh, it's getting good.
No.
Don't bring
I'm
up
not
crosswords because
joking.
Rachel will get one up
I
and we will
don't-
be here forever.
I don't know.
Guardian Daily Crossword. Shout
How
out.
many have you done today?
I actually haven't done one today.
Or are you even
I
Rachel?
know,
Are you
I know,
okay?
I know. I am okay. I've just been kind of
A bit, like-
dozing off. Yeah. It's just
Do you
been one
feel
of those days.
uneasy if you don't?
I
do it?
actually have been feeling really anxious today.
I was
But
about
don't
to
think
say.
it's the crossword.
I was about to say.
I feel like I can see how a crossword, doing your crosswords would be your little me time
It's
out
my
of my
safe
head.
space.
Yeah,
Yeah.
ritual,
Yeah. It's
I was like that.
grounded.
I used to be like that with Sudoku, and then I became like that with nonograms.
Okay.
And now I'm into those colouring apps, colouring games, where you do the colour by numbers. Anyway.
I will definitely do a crossword on the way home though, so it's okay.
Nice.
I know that most stations in South London have the Metro or the Evening Standard, so I'm going to be fine. I've got my pen.
So, wait, hang on. Question about your crosswords.
Please.
How come you find newspapers to do it and don't just get an app?
Because newspapers, I like the fact I'm not looking at my phone. Makes it way harder to cheat. Also, I find it just really nice that somebody has... Well, I don't know if this is even true, but I feel like somebody's taken the time out of their day to create this crossword for the nation. It's like an art form.
Yeah.
So the Guardian Daily Crossword will have the setter, so the person who's created the crossword of the day. And sometimes when I try cryptic crosswords, which are harder, the clue's more lateral thinking, I am better at certain people's cryptic crossword.
Oh,
So it's,
God.
yeah, I'm like learning the patterns. So I appreciate the crossword setters, the unspoken heroes of the world.
Oh
so
my God.
if you decide to
I
leave
love that.
science, you would be an excellent crossword setter because you have so much experience in them.
See, I don't know if I'd be any good writing them, but I could definitely do them. I think I'd probably end up being like, you know, on game shows like The Chase, like someone like that. I would be that person.
So my mum knew somebody who used to do the like Christmas crossword for the newspaper that we used to buy at home in Ireland, and like it was like her claim to fame.
That's amazing.
And he used to just do that crossword. I can't even remember what the relationship was, but he used to just do that crossword and he got paid quite a lot of money for it.
That's really
Because it was like
sweet.
the biggest crossword of the year.
Yeah, the
Was it
special
Christmas
one.
It was
themed?
big. No,
Okay. It just came out at Christmas.
it was just the like, you know, for all the crossword junkies such as Rachel. It was like, you know,
You
their
must
Christmas
be
present.
you must be excellent at pub quizzes.
Yeah, I love pub quizzes.
Okay,
So
I need to bring you to my next pub quiz.
Okay, I'm in.
Oh my god, I'm taking you to mine as well.
Okay, yeah, I'm in.
Should we just all go to one together?
Oh my god, yes.
Yes.
Yeah.
We'll
yes.
go to the tooting one. That's my fave.
Ooh, lovely. All right, I'm in.
Anywho, your PhD.
a brief interlude.
Where were we? Oh yeah, all it says is zebrafish at this point. Okay, so I'm looking at the optic tectum, and this part of the brain in the fish is really important for making movements where the body is kind of in the center of space and everything else is around it. So the word for that is egocentric movement. So like you are in the middle. And the kind of similar part to the optic tectum in the mammalian brain, in our brains, is called the superior colliculus. And it's really important for things like if you're reaching out to grab something, the hand movements involved in that. Or if your eyes are following something, if your gaze is tracking something, then that part of the brain is really important.
Does
for
that part
as
of the
well.
brain get fucked up when you're drunk?
I was about to ask what happens to this whole part of your brain when you're drunk.
You know what? I actually haven't thought about that, but I guess it kind of makes sense.
Have you
But
not been getting
also-
your zebrafish drunk? Rachel?
No, I haven't. Oh my god, I'm a good fish mama.
Oh wait, didn't you have a project where you were literally injecting alcohol into chicken eggs?
Okay. This is what I did as an undergrad, yes.
Oh my God.
So basically the idea was...
Oh, like fetal alcohol syndrome.
Exactly,
Yeah.
yes.
Oh.
I was trying to understand fetal alcohol syndrome, and the way that I was doing this was by injecting ethanol, which is the alcohol bit of alcohol, into a chicken egg. And the reason I was doing that is because in a chicken egg, the actual chicken bit develops on top of the yolk. So if you make a tiny little hole in the shell and then put a syringe through that, you actually won't like crack the egg. So you can introduce alcohol at a concentration that, you know, is similar to how it would be delivered to a bubba in the womb.
Oh
you
my
know,
God.
theoretically. I stained the brain to look at different types of neurons and see what was different basically after adding this alcohol. And the types of neurons that I was looking at is really simple, two really broad categories, just excitatory and inhibitory. So excitatory neurons tell their neighbor to fire, to send information, and inhibitory neurons will tell their neighbor to be quiet, basically. And what I found was that at the boundary in the developing fish brain, where you'd expect kind of a balance. So this area is called the midbrain, hindbrain boundary. So between the middle of the brain and the back of the brain, this balance between excitatory and inhibitory cells had been disrupted. And an excitation inhibition imbalance is what's been suggested might cause some of what we see in fetal alcohol syndrome. So ADHD, sleep disorders and intellectual disability, some of these things have been, it's been proposed that an imbalance in excitation and inhibition in the brain might make these things happen. So that's why I was putting alcohol in chicken embryos. Into eggs, yeah, with chicken embryos and little tiny ones. Yes. Where were we?
So your actual PhD.
My actual
Oh God, no, we keep getting sidetracked.
PhD.
Sorry.
It's okay. So my actual PhD is to use zebrafish when they are really young. They're just 6 days after being a fertilized embryo, so a blastocyst. And at this stage, they only have about 100,000 neurons in their brain, compared to humans where we have, you know,
Trillions.
freaking billions and gazillions, whatever.
Holy moly.
But they're still capable. Yeah, go on.
How big is a 6-day-old zebrafish?
About 4 millimeters.
Cute.
Oh my god.
Yes.
They're like little sea monkeys.
Yeah, they're really tiny. They're like, you know, if you looked at one, it's definitely no bigger than a little caterpillar or something. But they're still capable of really complex behaviors. So they can hunt food. And in fact, the way that we feed them is we drop some single-celled organisms into their petri dishes, so they swim around in the water. We drop in these organisms and they float around, and the fish will hunt them in the water and snap them up in their little mouths, and it's adorable.
Do you have to watch all this through a microscope?
I don't have to watch them eating, but somebody in my lab is looking at prey capture and, you know, can we understand how the fish captures its food, and then how can we relate this to brain circuitry? And yeah, some really interesting stuff is happening with zebrafish prey capture. But I don't have to watch that.
I mean, if you want to,
If
can
I
you?
want to, I can.
Because I feel like I would want to.
Yeah, and you can see them like.
Yeah,
so basically,
you
they'll
monster.
like.
What?
You're a monster.
Well, it's just like
munching.
it's pretty
They're
natural.
like microorganisms.
Yeah.
They've one
Yeah.
cell.
Yeah.
I'm just kidding.
And her
They're not
PhD
even a brain.
And Al's PhD is in snacking, and these are
They're just having a snack.
having a snack.
Yeah, and it's cute because you can see them just like swimming around, and then they'll notice a bit of food, and then they'll, you know, turn themselves to be near it, and then you'll see them like do a little jump forward, and then a big jump, and they'll eat the food. So it's really fun to watch. It's really lovely.
Zebrafish can learn, can't they?
I mean, I haven't looked too much into it, but yeah, it makes sense. I mean, what I should say is the zebrafish that I'm looking at still, yeah, super young. So I'm not looking at adult fish. And yeah, I assume there's been a lot more learning stuff in grown-ups than in larvae. So yeah, they have about 100,000 cells in their brain, but can still do all these fun things. And like I said, some of these behaviors are controlled by the tectum, which is the bit that I look in. So my specific question is, how does activity in the tectum generate these movements? And to try and understand this, I'm trying to see if there's a link between where the cell is in the tectum and, you know, the part of the motor network that it's connected to.
Right.
Because earlier experiments in goldfish show that if you stimulate the front part of the tectum, then you can generate different eye movements to if you stimulate the posterior part further back. So there's this idea that, you know, functions within the tectum are defined based on where you are. But we don't really know what these cells look like. Can they be classified into different cell types? Might that tell us about how the tectum works to bring together all of this information and make the right movements? So one really helpful thing about these larval zebrafish is that at 6 days post-fertilization, you can get some transgenic fish, so they've been altered genetically. And they are see-through, basically. They have no melanin, so the stuff that gives our skin colour is kind of the same for them, and they just don't have it. And this is great because then I can use their genetics to make certain cells glow in the brain.
Cool.
And image them in the whole fish without having to dissect anything. So that's why I use the zebrafish, and that's how I look at my cells in the
That's
tectum.
so cool.
That's really cool. And you know, when I looked up zebrafish, I saw a picture and it looked like I don't know what I thought it was like an embryo or something, but I think it was one of the light up see-through brain zebrafish.
Maybe, yeah.
That's going on the Instagram as well.
Instagram is going to be so full from this one
this is
episode.
just gonna-
So does that mean that you don't ever need to sacrifice your zebrafish?
Right, yeah. For the experiments, I don't have to sacrifice them, but basically for all animal research in the UK, it's really tightly regulated, and once you get to the end, you know, the most humane thing is to let them go.
Which is like, so I learned the other day that even with wild type mice and stuff, we still need to, in the UK, you still need to sacrifice them. Whereas I think countries like Germany have started letting you rehome wild type animals.
So I think it depends what they've been used for. And also, like if you use sacrifice or cult a mouse, it can be used in other research, again, depending on what it's been used for.
Yeah.
So if you use a mouse in a task that's all about learning, and then you sacrifice at the end of that experiment, you could then, you know, use the organs in research on cardiovascular stuff or whatever.
Yeah. That's so cool.
Yes, I like it.
Yeah. I don't even know what to ask. This is why we need a layperson. Ask a question.
about zebrafish. Oh my god, no, I feel like I'm too close to the research that I already know a lot about.
But I also feel like you've explained that really well.
You have.
You've explained that really well, because I think that I think there probably are people that wouldn't explain that so well, because it is quite I don't know if abstract's the right word, but like
it's a bit abstract.
I think with work like that, it is really hard to relate it back to humans. Okay, so say as a model, what, apart from those benefits that you're talking about with zebrafish, like one, they don't live like they're quite quick to breed, so you can have lots of experiments quickly. Two, you can do the whole genetic manipulation things, they're see-through. I'm giving you like a mini Viva here.
Yeah.
So they're see-through and like their brain glows, but compared to say a rodent versus human, they're extremely different, right?
Yeah, this is true. And I think this is another thing about pure research and why do we do it and how is it relevant. I think what we're really trying to understand, or at least from my perspective, is the principles that underpin how a brain is able to work. So my work about how do these different areas connect, you know, all the work that happens in my lab kind of complements each other. So there are other people looking specifically at activity in these cells. So not only can I make the cells glow kind of permanently to look at them, we can make it so they light up more when they're active. And because you don't have to, you know, like cut any brains, you can record a video of this activity and understand which cells are active at which time, and how does that link up to maybe movements that the fish is making in response to something that looks like prey or something that looks like a predator. So you can show videos and look at activity, or you can just look at activity when nothing is happening and understand which types of cells are naturally, you know, working together. And if we understand that, and then look at what these cells look like, what brain areas do they project to, then it could tell us functionally how, you know, the brain works together as a bunch of really cool circuits, and maybe one massive circuit all working together to do all the things that we do. So then the important stuff is not, okay, how does this part of the fish brain connect to this part of the fish brain? An important thing could be, we know that a circuit like this can exist. Do we see anything similar in humans? Even if it's in a completely different place, you know, because then it would let us know kind of how that circuit might work. Or we might be able to find some specific like pattern in activity that maybe we see during epilepsy in a human, because an epilepsy would be like, you know, this kind of wave of activity across the brain. There are epilepsy models in zebrafish as well, so I guess that's more of a direct link. But yeah, I guess that is how I would put it all together.
Tristan, chime in, because same with you, the animal models that you were using are so different from mammals that we would normally expect to be used as a proxy for human.
But yeah, I think I agree with Rachel. Like, if the whole thing about looking for conserved structures and pathways is the whole reason we use models. And I think with my research specifically, it would have been really difficult to kind of look at how foliation develops in just folded brains, because that's not gonna really tell you anything. Like if you look at a folded brain and you look at a non-folded brain, it's very easy there if you compare their developments to be like, oh, that's different and that's different. And then in our brain, it's a folded brain. And so if we see how a brain folds in a bird, that's already going to give you some clues as to how our brain folds. So it's stuff like that. That's why models are important.
Yeah. And just to go back to, you said things that are highly conserved.
Yes.
Conserved.
Conserved is not a layperson word. I don't know why. I don't know where that came from.
No, Tristan, you let the guard down. the lay guard.
I think you might not be a lay.
I'm skeptical.
Unlay him.
I'm such a lay. But yes, conserved. You asked me to explain conserved. Conserved basically throughout evolution as, oh my god, now it's sometimes really difficult for me to get to be not technical about these topics. But yeah, throughout evolution, as different species have kind of split off from each other and have become different, certain things stay the same, and that's what we mean by conserved, basically. The thing, like structures, like the brain is a conserved structure.
Everybody has a brain.
Everybody has a brain, like your deer.
All mammals have...
Insects.
Insects.
Yeah, all those things have brains. So that's what conserved means.
Yes. Just so you know, I knew what conserved meant.
I know you're not a lay person. I didn't know what conserved means because I'm a lay person.
No, you didn't know. You didn't know.
Yeah.
No, that was someone speaking through you.
I'm actually a ventriloquist.
She's very talented.
Rachel was just throwing her voice into Tristan.
Yep. And now I'm informed. Now I know what conserved is all about.
You're welcome.
It's so interesting because I think we've had a lot of science guests on now, but you guys are so different
Yeah,
from what we
it's
do.
super interesting.
Because all the other science guests we've had have been pretty similar to what we do.
Yeah, either hippocampal neurogenesis or...
That's not true. We haven't had a single guest on who does hippocampal neurogenesis.
Have we not?
No.
Demelza. She's not a guest.
She's not a guest.
Sorry, Demelza. Okay,
Yeah,
fine.
You should have more basic bitches on.
We should.
I mean, I love basic.
Do you know what though? If we should bring on Demelza should talk about her work because she does basic science.
What does she do?
She looks at the impact of prolactin and estrogen and testosterone on hippocampal neurogenesis and the impact that might have on postpartum depression. But she's now more focused on the actual
Depression-y bit.
the mechanisms rather than because the depression thing sort of went.
You know what else is interesting about hippocampus? The sleep wave stuff, the replay. Hippocampal replay, have you heard? Okay, cool. So there's basically, it's been observed that while you're sleeping, cells in the hippocampus will basically replay patterns of activity that have happened during waking. And that's some of the hypothesis about how we learn, because the hippocampus is really important for learning and memory. So yeah, it's very basic stuff where you're recording from individual neurons or groups of neurons and looking for that activity and seeing, if we see all of that happen, does that make them any better at a learning task or something like that? So yeah.
Is that related to how we dream at all?
So yes, some people have said maybe, so alongside doing all my PhD science stuff, I've also met a lot of really interesting people who do very different sciencey stuff by doing science communication and weird events and stuff. And I met somebody who researches lucid dreaming, and she talked about some of the ways in which people use lucid dreaming. So
I've done it a few times.
Yeah.
before.
I did it and it was fucking terrifying.
Really?
Like, it was a nightmare. I was
Is
like,
anybody watching The Haunting of Hill House?
Yes. Wait, I'm on episode 3. Don't say
no
anything.
spoilers, please.
Wait, what episode are you on?
all of them done.
Oh, you
Done.
finished. Okay, I'm on the
I'm getting
8th.
excited.
I'm on the 8th. But her lucid dreams are
Wild.
wild.
Oh my god.
Wild.
Yes.
I would be skeptical on if they are actually lucid dreams, but they look terrifying.
No comment.
What is her hippocampus doing? Do they address that on the show?
They haven't addressed it yet, but I have 2 more episodes to go, so
Okay.
here's hoping.
That's definitely
it's coming up.
in
Yeah,
this
I can't wait. That's where I thought it was going. That's the track that they're on.
Straight to MRI.
Straight to the MRI. All about the hippocampus. Such scientists.
What were we talking about?
Lucid
before
dreaming.
this?
You met somebody who
Yeah.
was
So a
researching
lucid dream, all it is a dream where you know that you're dreaming. And some people think, oh, if you're lucid dreaming, you have to be able to control everything and fly away. But no, it's just a dream where you know that you're dreaming. But yeah, if you know you're dreaming, then you can, learn to use it. And there are different techniques to do that. But she basically talked about how some people use lucid dreaming to try and reinforce things that they've learned when they're awake. And there's like some very, tenuous stuff that shows that maybe, it could improve. Like, I think some people had tried to see if people improve at a learning task after practicing while lucid dreaming. I didn't look up the study too
How
closely.
did they know that they were
or anything.
lucid dreaming though?
This is the cool bit. Okay, so they have people do 2 kinds of not tests, but 2 different recordings. And the first is an EEG, which I guess you probably talked about. What was it? Electroencephalogram, where they record electrical signals that are just dancing across your skull from your brain activity. And then the second recording they take is an muscleogram, basically. I don't remember the proper name. So EEMG. And they record the movements of the eye muscles, or the signals being sent to the eye muscles. So they tell people when you are dreaming, once you know you're dreaming, go on.
Ding, ding, ding.
Yeah.
True fact.
Oh, God.
Electromyography.
Thank you. So they say when you know that you're dreaming, move your eyes, really strictly left, right. So then,
Oh, shit.
they look at the EEG, they look at the brain waves, and they're showing that they're asleep. And then they look at the freaking EMG, and they see left, right, and they know they're lucid dreaming.
That's cool.
God.
Cool.
That is really cool.
That is so cool. But
yeah.
also, how do they actually know that they're lucid dreaming and they're not because at some point during the day they must have gone left, right. And what if that's just kind of muscle memory happening in your sleep?
So I think again it comes back to this rapid eye movement, and I might now be getting this wrong, but I think dreaming happens during rapid eye movement, which is very different to the left, right that they've told them to do. So they can tell the difference. And if you look at the traces, they actually look mad different. So it's legit.
Oh my god, that's cool.
That's crazy.
Yeah.
And when thinking about that research, they probably had to have those people living in a sleep facility for months to get all that data. Because it doesn't happen that often, does it?
And I bet they got paid a fuck ton.
Oh, shit, yeah.
Yeah.
So there are techniques you can do to make yourself lucid dream.
Yeah.
This is just a massive tangent that I've gone on. So thanks for
Do you know what? A
tolerating
part of me is should we even bother talking about your paper? 'Cause
I know, we've covered
we're having
such
such good discussion.
Yeah.
interesting things.
Yeah.
Yeah, paper's not the shining star. I mean, it's all right. I'll
Why don't you give us a
Give
really
us a
quick-
hot fact.
Yeah.
Okay, so I chose this paper because it scared the shit out of me.
More than The Haunting of Hill House.
More than the Haunting of Hill House. More than watching the entire season within the span of 1 second. I saw it and I was like, oh my god, am I crying already? I haven't even taken it in. So basically, my goal at the moment is to image as many cells going out of the tectum as possible. And in this paper, a bunch of different labs have collaborated to label over 2,000 cells in the zebrafish brain. So literally when I saw this, I was like, I'm dead. everything I've done means nothing and my life is over. But then I read it and I was like, you guys aren't looking at what I'm looking at. And also, I figured, if I can get a good number of my cells and add it to this database, that's what science is all about.
Yeah.
Then I'm helping out everyone. Open science
Collaboration.
for the win. So yeah, I read it and I was like, oh, this is kind of cool. And it was also really nice because it showed me that I'm on the right track, because I'm doing similar stuff and using similar logic. And it basically reflects this whole thing that's going on at the moment where people are trying to make a lot of different atlases.
Very trendy.
Yeah. So the same way, an atlas tells you where different parts of the world are, a brain atlas tells you where different parts of the brain are. And, the special thing about this atlas is it is at single cell resolution. So like the first typical way of understanding parts of the brain came from this guy called Brodmann, and he had his 32 or 36 areas. So that's really large scale stuff. Like these big folds here are the temporal lobe.
Yeah.
And but this is single cells in the zebrafish brain looking at 2,000 out of 100,000 total. But still, that's a lot of cells. To my knowledge, the most that anybody's ever got kind of into one beautiful brain atlas. So what they've done is they've talked a bit about how they made this atlas, and it kind of talks through more methods that I use in the lab as well and why they're interesting. And then it tries to understand principles of how the zebrafish brain connects together and works together, and talks about why atlases are cool and shit. So that's the paper. But I mean, I'm also happy to just carry on chatting. It's up to you. It's your podcast.
I mean,
don't know.
we're 1 minute over time.
Yeah, we've actually made really good time.
We have.
Excellent.
Yeah.
All right. Well.
And I
Great.
feel like we've covered a lot of
Yeah. I
science.
mean,
have.
we've got it all.
And
It's all in there.
such a new, interesting area
Yeah.
for
I think-
the podcast,
I think
not
if-
for the world.
I think if I was a lay listening to this episode, I've had a lot of
me.
information.
Yeah.
I also love how we shortened it to lay.
I also
if
like
was
how
lay.
our lay guest asked the least amount of questions.
I was just too overwhelmed by how ununderstandable everything was.
You were just too late.
Yeah, I was too late.
I just didn't get it.
I just didn't get it.
Jonah, let's get you back to your lay. Should we do a question out the cup?
Oh,
We've got a little bit of time.
What
I suppose we do.
is that?
We can always cut it out if it's shite,
Yeah.
which it sometimes
What's
is.
the question out of the cup?
So,
So the
I'm
cup
gonna-
is the cup of questions.
Hopefully it's a good one. Ooh, okay. Question 10. Just because this podcast has been quite highbrow, just to lower the tone a little bit.
Yes. Bring it down.
What is your favourite curse word?
God.
Mine would
so
be
many.
mine is probably fuck.
Sorry, I'm thinking.
Yeah,
I like-
feeling?
I also like motherfucker. I say that quite a lot.
You do. You really do, yeah.
Yeah, it's when I hurt myself, it's my response swear word.
I kind of like, I like it when old Irish men say feck a lot because I find that hilarious.
That's nice.
Just to be
Oh,
stereotypical.
shit. Yeah, if we're going regional. Then.
Regional.
I also like
Sticking with
bollocks.
heritage.
It's not really that bad of a word, but I'm just, when you do
Bollocks.
something, you're just like, bollocks.
I like bollocks too.
Yeah.
Bollocks is a
I
good
think
one.
English swear words are really satisfying.
But English swear words are still so polite. Like I quite like prick because I feel like it sounds quite,
The thing is, have you seen those
absolute.
videos?
There's a prick. There's a bit of onomatopoeia in there, isn't there? Prick.
Yeah.
I think that's what's good about fuck though, because it's like, fuck. And
I see
also,
what you mean.
fuck can be used in any situation. It can be used for any emotion.
It's
Yeah.
very versatile.
Yeah.
I feel
It is
like
versatile.
The thing is, though, to me, English swear words almost don't feel like swear words because if I feel like I was really mad, I'll go straight to Jamaican patois.
Oh.
They're like swear words.
Give us a sampling.
Oh no!
Pretend that we pissed you off, Rachel.
That's the thing. I feel bad saying them.
Oh.
The thing is, okay, I have a theory about this because I feel bad using Korean swear words. I never say Korean swear words because they feel so bad.
Yeah.
But I wonder, okay, here's a theory. Here's a psychological theory. I wonder if it's because for people that grew up here, in terms of those swear words, so we would have only really associated them with our parents,
Yeah.
or like, and you would never speak like that in front of your parents. So I wonder if that's why we feel uncomfortable saying them, because with our English swear words, we're saying them with all our friends. But I would never ever use the Korean swear words, because the only people that I would use them with is my parents.
I think that's it. That's definitely it. And especially as well when somebody doesn't know what a swear word means, and then you have to explain it.
Yeah.
Loses
Then you're
a
like,
lot of its
not
power.
loses it. You realise just how strong it is.
Yeah.
Maybe.
So yeah, there's one that is like the worst. It's basically I won't say an accent, I'll just say it in my shit
It's
London
so strong
accent.
you can't even say
I
it.
cannot say it in that pussy clot.
Oh yeah.
And what that means is, okay, the clot comes from blood clot, which is a blood clot, and then the pussy is pussy.
Yeah.
So it's like basically a clot from the period. Like, that's fucking deep.
A period.
That's
clot.
beyond. Sorry. Yeah, exactly.
Ooh.
Yeah, exactly.
God,
That's
that
bad.
is savage.
Exactly.
And now, would you use that to describe a person, or
Yeah.
and would it usually be used to describe a woman, or can it be used to describe
absolutely
a man?
gender neutral.
Anyone.
Oh, nice.
Yeah.
Nice. I like it.
I like dickweed. I don't think I don't think it's even an actual swear word, but I've made it up, and I like every time I'm pissed off with someone, I'm like, they're just fucking dickweed.
I like that.
That's beautiful.
It is.
I don't even know what that even means. I'm trying
But
to
you know
think
what?
about
I
it.
feel
It's-
like we wouldn't. It doesn't sound as good with a British accent though.
No.
Oh yeah, maybe not.
Dickweed.
Dickweed.
That was the most South London.
Dickweed. It sounds like you're referring to someone who's called Richard
Dick
Weed,
Weed
and it's like,
again.
oh, it's just dickweed.
Do you know what's best though? The best thing about English swearing is, have you watched those like near car accident, like those dash cam videos? And whenever it's an English man behind the wheel swearing, it's fucking fantastic. Because anyone else will just be like, oh, fucking motherfucker. But an English person will be like, you fucking wanking twat of bollocks, blah, blah,
Ooh,
blah.
twat. I love twat.
It's so good.
But another thing I like about British swearing that I feel like is quite British is really like longing it out to just like express how tired you are. Like, fucking hell, it's not the same as fucking hell.
Yeah.
That's what feels good.
Yeah.
It does.
Yeah, I don't think it would work in my accent, so I'm not even gonna. No.
Okay.
Where's your accent
Fuck
from?
it, no, it doesn't work. I don't know. It's just a mishmash of all of the places.
Yeah, because you don't sound very Filipino.
No, and
No.
but it's not really discernible. Like, tell us now where is the mishmash?
The mishmash. So I grew up in the UAE, but to Filipino parents, and apparently I had an Indian English accent for a while because my international school was run mostly by Indians.
Okay.
So I had that for a while,
Yeah.
and then I came here to Stoke-on-Trent, which is in the Midlands. It's where
gosh.
Alton Towers is, if anybody
Yeah.
Oh, fun.
I've
is wondering.
been.
Yeah. They have a very particular accent.
Yeah.
The first day, so I landed, and then the next day I had to go to school, and then the teacher was like, this guy, and from that I gathered that this guy was going to show me around, and he was showing me around, like pointing at things, and I was like, I don't understand what you're saying.
Oh, no.
But then like I couldn't really adopt that accent, and so I don't know, I kept this. It sounds a bit American, but then also not at times. It gets really American if I'm in America.
Actually, I can kind of hear the UEA-ness.
What do you
Or the
mean?
international.
Yeah, it's definitely an international
Yeah.
school
International
accent.
school.
It's just an international accent,
Yeah.
I think.
Guys, I have a story about Alton Towers.
Please.
So, I had laser eye surgery, so I used to wear glasses. And do it. It's the best decision I've ever made.
She describes having an epiphany while looking upwards in the rain.
Yeah, I actually did.
She was like, "Whoa!"
I
this
did.
is what it looks like.
I was walking home and it was raining, but it wasn't like heavy rain. It was like that, like the cloud of spit.
Okay.
Yeah, it was like that. And I was walking with my head down, and then I was like, shit, no, hang on, I can look up. So I looked up and I looked out, and it was nighttime, and the street lights were on, and it was just so beautiful. And I was like, oh my god,
That's a beautiful.
I literally
story.
had a
quite
moment.
deep,
Yeah.
isn't
I
it?
had a moment. I was like, I've never looked up in the rain before.
It is quite like that's that's
When you
nice.
said look up the first time, I imagined like actually looking up into the sky and like the rainwater just like, filling your eyeballs, and it's like, oh, what? But okay, this is much
My
nicer.
eyeballs.
No, okay, sorry. I looked ahead.
To a new life.
Yeah. Anyway, my Alton Tower story. So my automatic reaction then was to take my glasses off before every ride, right?
Yes.
So then we went on Nemesis, but they have two Nemesises. They have the Nemesis, which is the roller coaster, and they have a Nemesis, but I can't remember what it's called. There's another bit to it. So I got it. I took my glasses off. I gave my bag in, and then we're all there. And then we get in this elevator, and then it starts moving, and then we realise this isn't a fucking roller coaster. It's like one of those horror experience mazes.
And then this is what inspired you to get laser eyes.
But you know what?
God.
The thing is, everyone around me was screaming because things were like touching you and stuff, and I was screaming, and everyone was like, "That was so scary." I was like, "Okay, imagine how scary it is when you can't see what the fuck is going on." And what pissed me off the most was when I handed my bag and my glasses in to the cloakroom. They didn't say, "Oh no, don't worry, you don't need to hand us your glasses." They just took them.
Not totally obvious, isn't it? And here comes another, one doesn't realise what she's in for. Or maybe they just thought you could see without your glasses.
Maybe.
Yeah.
They probably thought I wasn't that blind.
Yeah.
Or maybe they were like, she's just one of those hipsters just wearing
Yeah.
for style reasons. She doesn't need them.
No, guys,
I have no glass.
I was very blind. I had no idea what everyone was screaming at.
Oh, God.
But you were scared anyway.
And I could just feel things happening.
That's even more terrifying.
Exactly. And
That is fucking terrible.
And I needed people to like guide me around. It was fucking awful.
You should have
That's
let
so
feedback
scary.
be like, if you want to make this scarier, just
Take your glasses
turn the lights off.
Oh yeah. It was fucking awful. Anyway. Should we end it there? On that note.
How do we all feel? I think that's a great note to end it on. Go out on a laugh and you being terrified and blind.
But not anymore because you can look up at the rain.
I can.
Her life has changed.
Do you know what else as well?
Feel the rain on your skin.
Feel it for you, only you can let it in.
This is the first singing we've had on the podcast.
It is.
Yeah, we've never sang before. That was beautiful, guys.
Amazing.
Beautiful.
Right, okay, that's the end of the podcast. Yeah, okay. So, did you guys have fun?
No.
Oh.
Yeah, of course I have.
Terrible experience.
You're the
I'm kidding.
first
That was
person to
really
say that.
good.
You're not coming back.
No, I did have fun.
Yeah.
I really did.
I felt very lay at points. So thank you for affirming my dream.
At points. Anytime.
I won't lie to you, I think me and Alice were more late than
I
you.
think we were more like.
But I tried. I'm a master's student now. This is what I'm a lay person. I'm back.
Oh shit, we didn't actually talk about your masters, did we?
Yeah, and also really interesting. It's only occurred to me now that you're doing a master's after your PhD.
Yeah,
Is that very common?
It's my path to layness. I just will go back.
It's a master's in lay.
No, so it's a master's in science communication.
That's
So
actually
it's
the
like,
only
you
reason
know.
why I let you come on the podcast. Because you're a future
Sure, it was
contact
like,
for us.
okay, we'll let him come on.
Working.
Yeah.
Yeah, no, I was thinking
How are we
about
doing?
that.
Very good, very good. I will take your business cards after. No, I'm kidding.
Shit.
Business card.
We should.
Oh my god, that'd be cool.
But yeah, okay, well, thank you very much to our guests. You've been amazing.
Did you have a good time?
Yeah, I had
Such
such,
an
a fun
awesome
time.
time.
Yeah.
Thanks so much for having us.
Yeah.
Thank you for
Thank you
coming.
for being our
Thank
guest.
you for making dinner for us and stuff.
Yeah.
You're welcome. Thank
was
you for all the
delicious.
snacks and the mulled wine.
The
First-
mulled wine was so tasty.
First mulled wine of the year.
There you go.
Oh my god.
It's
And it's,
basically Christmas now.
Yeah, it will be
Basically.
when this goes out.
Yeah.
I mean, it's pretty much November. It's fine.
yeah. I mean, it will be November when this goes out.
Oh my god.
it's in through-
It's
yeah.
almost November. We can put up our Christmas decorations. I'm sorry, none of you live with me, but I'm just saying that to myself.
But yeah, if you would like to, you can follow us on Twitter and Instagram on @layoutpodcast.
You can email us on layoutpodcast@gmail.com.
You can read our blog by Silent Demelza on layoutpodcast.wordpress.com. It is a bit behind.
You say that every week.
No, because
they know it's behind.
I know, but we might have new people. It's a bit behind because Demelza is coming up to the end of her PhD and is starting to write her thesis, which is a time when people do not live normal lives.
No.
So
Can definitely attest to
Says
that.
the lay person.
I don't know what you're talking about right now, I'm very late.
I'm so late. Okay.
okay, bye.
Goodbye.
Bye! What is your book idea?
So. And not Not too much crisp munching. None of that.
Curie gets really angry about things. We can just laugh at her.
Oh my god, I'm such a layman right now.